All right, so it's 1 o'clock. I'm going to go ahead and get started. I'm really excited to introduce our March seminar speaker, Dr. Vincent Reynolds. He's going to be talking today. The title of his presentation is The Threshold of Toxicological Concern as a Risk Management Tool. Dr. Reynolds is a toxicologist with over 3 decades of experience in the chemical and pharmaceutical industries. He's completed his bachelor's degree at the University of Delaware and PhD at the University of Kentucky and postdoctoral studies at Fox Chase Cancer Center in Philadelphia. He spent 12 years as a toxicologist in the chemical industry, where his activities support workplace exposure guidelines and environmental remediation and development and reproductive toxicity assessments of commodity chemicals and consumer product components. In addition to his activities as a toxicologist, Dr. Reynolds has been active in the fire service since 1973 as a firefighter, EMT, engine chauffeur, ladder chauffeur, and tillerman, and field instructor. He holds certifications as an emergency medical technician, emergency vehicle operator, firefighter 1 and 2, fire instructor 1, 2, and 3, fire officer and safety officer, and he was an active field instructor with the Delaware State Fire School and is currently a life member of the Aetna Hose, Hook, and Ladder Company in Newark, Delaware, and a lieutenant in the Greenfield Fire Department in Indiana. So without further ado, I'm really excited for this talk. I know many of you guys have been lucky enough to meet Dr. Reynolds at our symposium, and we are lucky enough for him to be on our team and to present today. So without further ado, it's all yours, Dr. Reynolds.
Thank you so much, Natasha. I wish my mother were alive to hear the very flattering introduction you gave me. She would have been proud. The title, as Natasha said, is The Threshold of Toxicological Concern as a Risk Management Tool. And what I hope to accomplish here this morning with you is to communicate to you a way of thinking about problem solving that may be really quite alien or quite foreign to many of you. For those of you who are in the fire service, who are used to making decisions where there are very limited data available, you have to make a decision where there's very, very limited information. This may seem somewhat similar, but for those who are more in the scientific realm, who are used to operating off relatively robust data sets to drive decisions, at least at first blush, this TTC, or the threshold of toxicological concern, might seem to be a little bit, as I said, alien or foreign. So what is it? The TTC is a tool that has been developed over a number of years that allows toxicologists and risk managers to make decisions on how much of a chemical we can safely take in, even if that chemical has not been studied for its adverse effects. And that sort of seems a little odd, but I'll walk you through how this is done. I want to start by describing for you sort of the internal landscape, or what the pressures are inside a pharmaceutical company as they're trying to develop drugs. And you'll see how these pressures come to bear and drive the decision-making process. And hopefully that'll help you understand why the TTC is so important. Then I'll take a few moments and talk about the external landscape, which is the regulatory framework which has sprung up to assist companies in developing drugs. And then we'll get into the TTC itself. Where does it come from? How does it work? Where does it fit? And in what situations would it not be appropriate to use a TTC approach? So the internal landscape of drug development, there are 3 pressures that I want to mention here. One is speed to patient. When we work on developing a drug, the clinical benefits that we're trying to bring to patients are to help them address unmet medical needs. So in many cases, the patients are either dying or they are suffering from really very, very serious medical conditions. And we want to speed things along as quickly as we can. And what that translates to is we don't have a whole lot of time to shilly-shally around, wringing our hands indecisively. We have to make decisions. We have to either push things forward or we have to terminate development and reassign the assets on a particular project to some other project where there would be a greater chance of ultimate success. So, you know, just standing still is not, that's not part of the plan. The other part of speed to patient is just simple business survival. I show here sort of an arrow, the green arrow from left to right, showing, you know, the process of drug development. It begins with discovery. Then there's some development activities, both non-clinically and clinically. At some point, it becomes available to patients. And I show that with the little marketing authorization box at the top. Once we receive that, the drug can be marketed and made available to the patient population at large. Initially, it's sold under a brand, and then eventually the generic companies come in and the drugs go generic. Now, the silver or the gray arrow underneath the green arrow shows the patent life. Somewhere in the early days of the discovery process, the patent is filed. And you can see the little gray box there for patent filing. The patent has to be filed before the drug leaves our property so that we have our intellectual property rights protected. That's just a given. That's the rules of the game. Once that patent is filed, it has a finite lifetime. It lasts for 19 years. And so you can see that the patent life actually starts running while the drug is still in discovery and all through development. The marketing authorization, as I said, when the drug becomes available to patients, is sort of on the latter part of that 19-year patent life. And that's the only time that the drug company can actually make money. So looking at the text sort of in the lower left-hand corner of the slide, the profit life for a drug is the patent life, that is 19 years, minus the time that it has been sitting in either discovery or development. So what does that mean? That means if you have $1 billion a year drug, that works out to $2.7 million per day. So every extra day that we spend in discovery and development before filing the marketing authorization and getting permission to sell the drug, every one of those days for $1 billion a year drug costs $2.7 million off the profit life. So, you know, you just don't have the ability to stand still in drug development. You have to make decisions. You have to drive things forward. So that you don't chew into that profit life. For drugs that are more than $1 billion a year, obviously, you know, the costs for development delays are even greater. And I have been on programs where we've encountered delays that were long enough that the business case could not be made to continue development. So this is a very real pressure that we feel, not only for the patient, you know, getting the medicine, but also for keeping our business, you know, profitable and healthy so that we can continue to discover new medicines. The other pressures that we have are safety and efficacy. The drugs have to be safe. They have to be efficacious, and those qualities are mandated by U.S. law. Safety, as we see, is multifaceted, and there may be some eyes that are opened by one of the safety issues that I'll be talking on here later in this seminar. And the other thing which people really don't appreciate, I think, is how closely the safety and efficacy of drugs in development are scrutinized, not only by the regulatory agencies, but also by the insurance companies or by the payers. And what this means is there's intense regulatory oversight all throughout drug development. The regulatory climate that the pharmaceutical companies operate in, and this is probably very, this might be new information to many of you, since if you don't work in this area, you might not be familiar with this. But the pharmaceutical companies worldwide, that is in the United States, in the European Union, and in Japan, have partnered in a collaborative interaction with regulatory agencies in those regions in an organization called the ICH, the International Conference on Harmonization. This is a global regulatory framework for pharmaceutical development and approval. And it was necessary because in the old days, there would be a lot of companies operating in different countries throughout the world, and each country had their own requirements. And it got to be very confusing because you could be approved in one country and not in another. And it just got to be a real mess. So industry and the regulatory agencies got together. They collaborated to come up with a framework to ensure quality, safety, and efficacy. Those were the three pillars that the ICH was built upon: quality, safety, and efficacy. They wanted to harmonize drug technical requirements so that one set of rules would apply throughout the world. And they wanted to avoid duplication of testing in humans, as well as unnecessary testing in animals. And you can see in the table there the different agencies from the regulatory side and from the industrial side that are part of the ICH. Now, why is this important? The ICH has come up with guidelines, and I'm going to start with the safety guidelines, the second column there. That encompasses the toxicology work, which is mostly where I've operated in my career. The efficacy guidelines are more on the clinical trial side of drug development. So that would be the human studies. The multidisciplinary guidelines cover topics that sort of branch into different areas. But what I really want to focus on here is the quality guidelines. As I said earlier, I think most people are aware, at least to some extent, of the importance of safety in drug development. And they think, generally speaking, about the toxicology studies that are done to support drug development, or the safety measures that are included in the conduct of clinical trials. What very few people are aware of is just how absolutely stringent the safety and quality requirements are for the drug product or the drug substance itself that is going to be studied and hopefully developed into a marketable drug someday. So there are a series of quality guidelines there in the first column, which define in great detail what the regulatory expectations are for understanding the nature of the molecule that's going to be advanced in testing. There are release specifications for each lot, and by that I mean all of the degradants and all of the impurities have to be controlled within certain levels And they're specified, and tests have to be done to demonstrate for each of those impurities and each of those degradants that you are below the specification limit. There are stability studies. There's a lot of risk management that goes into the manufacturing process. That is a world unto itself. And that's the world where we're going to be moving here with these next few slides. This, so we talk here about impurities and degradants. On this slide, I'll introduce you to the ICH-M7 and the Q guidelines. The Q is for quality. And if you have, if you want to learn more about this, you could just Google up the ICH and you can look at these guidelines. They probably have more information than what you might think and what you might necessarily need. They're quite detailed and they're quite lengthy. But these guidelines from ICH, the quality and the M7 guidelines, we need to assess risks for all detected and potential impurities. So it's not just the ones which we can see and measure, it's the ones which might be there. And in the two large boxes with the orange border, these are synthetic pathways from a drug that I worked on a number of years ago. And you can see that these synthetic pathways can be quite lengthy. I had one program one time where it was a 19-month synthesis. It took 19 months to make the drug. Quite a few steps. This was a much simpler synthetic route here that we're looking at in these two boxes. And you can see in the fine print there are a lot of catalysts, there are a lot of reagents and solvents, there are different starting materials, there are reactive chemicals, there are processing aids, et cetera, that sort of lurk throughout the synthetic process. And these all have to be controlled. If there is a change in the synthetic route, then the assessment has to be repeated. If there's a change in the formulation or the composition, if we do a salt change, if we mill to the product so that we have a smaller particle size to enhance absorption, if we change the salt form so that we get better stability, any of those changes, we have to go back and go through the whole assessment process over again. This is very, very, very thorough assessment, which is needed. If we change the indication so that there's a change in the dosing regimen, that changes the assessments that would have to do under the M7 and the Q guidelines. So there's a staggering amount of work that goes into this just so we understand the nature of the molecule, the nature of the drug that's going to be tested. And as I said initially, these assessments have to be done quickly. We have to be able to reach decisions in a reasonable time frame so that we don't delay getting the drug to the patients who are in need, and that we don't basically run out the clock on the patent life and destroy our ability to stay in business. So there's actually organizations within pharma whose sole job it is to handle the test article characterization work that we've been talking about here. So that moves us to the question, if we look back here, many of these intermediates or starting materials or reagents or degradants or side products or byproducts in these chemical reactions may not have been studied. There may be little or no toxicology information at all available on them. And in fact, many of the intermediates themselves would not have been studied. They would have never been synthesized before, except for use in one or another of these synthetic route pathways. So there would not be any information to tell you what the toxicity properties are, what the stability is, and so on and so forth. So here we come along and we have to assess the risks for these compounds when we have little or no data, because some of these compounds may trickle through at very low levels and find themselves in the final drug substance that's going to go into the clinical trials. Okay, so it's not always possible to remove 100% of the impurities prior to administering the test material to humans. And how do we assess risk for that? So what we did in pharma was we reached out into the broader scientific community, where in the food world, some scientists were trying to come to a better understanding of how much of a food additive could be present in a food product without creating any adverse health effects. And so way back in the 1970s, and the reference is down here at the bottom of the slide, Kramer came up with a classification scheme, which is largely in use today. He divided the world of food additives up into 3 separate classes. The first, class 1, these were very simple chemical structures with known metabolic pathways, with end products that were known or very likely to be essentially innocuous, and therefore would have a low order of oral toxicity. And he did have, in many cases for these, actual toxicology data to support the classification of compounds here in class 1. The class 3 compounds had chemical structures which did not give you a warm and fuzzy feeling, did not convey a sense of safety, and that had structures which might not only suggest, but might be indicative of toxicities. And then the class 2 were somewhat in between. They were not as alarming as class 3, but they were certainly less innocuous than class 1. And for these 3 classes, Kramer proposed what he called the threshold of toxicological concern. His proposal was that compounds, say in class 1, could be present in food so that your oral intake could be as high as 1,800 micrograms per day, or almost 2 milligrams per day, without any risk of adverse effect, any risk of toxicity. If you were in class 2, you could have an oral intake of 540 micrograms per day, and for class 3, the threshold of toxicological concern was 90 micrograms per day. And you can convert that to micrograms per kilogram per day, if you wanted to adjust it based on body weight. But generally speaking, you could just say, the 1,800, the 540, and the 90 microgram per day TTCs would apply. This was not, these values were not arrived at arbitrarily. One of the things that Kramer did as part of his analysis was he looked at sort of a very broad range of compounds that had been tested in toxicology studies. And what I show here is a dose response curve. It's a very simple diagram the dose is on the x-axis. So as you go from left to right on the x-axis, the doses increase. And then the response, whatever the toxicity response is, whether it's liver toxicity or neurotoxicity or death, whatever it is, whatever the endpoint is shown on the y-axis. And the numbers that I show there are just for illustration. They're sort of an imaginary dose response curve. But if you took the dose response curves for a very, very, very large number of chemicals that had toxicity data generated, you would find that his TTCs were at the extreme lower end of the dose response curve. So they were way, way, way down in a dose range where there would be little or no likelihood of any adverse effects. And the NOAELs, or the no adverse effect levels for these different chemicals that were part of the evaluation, were identified, and then uncertainty factors were added to reduce the TTC even further to get down to the values that Kramer proposed. And I don't want to get into uncertainty factors in this presentation today, but if you're interested in that, you can contact me. My email was on the first slide, or you could see this reference here that I give by Dankovic. And Michael Dewerson was part of that paper, and he has been in the uncertainty factors world for a long time. But basically, these are ways to account for data limitations. Basically, you take the animal data and you identify, say, an NOAEL, and then you can reduce that by 10-fold or 100-fold or 1,000-fold to get to a human allowable intake, using these uncertainty factors. So it's a way to buffer oneself against the uncertainty that we have when data are limited. Okay. It's a fairly common tool in risk assessment. So Kramer's TTCs had been sort of operating in the background. They were not really part of pharma until the ICH guidelines came out and people began to see the importance of characterizing the test articles and the drug substances that were going to be advanced into human clinical trials. And with that, the concept of the TTCs was adopted or imported out of the food world and into the pharma world, where it's been developed quite a bit beyond what Kramer originally envisioned. So what about, I'm going to focus just specifically here now on mutagens and carcinogens. The TTCs have been applied to a very broad range of solvents and intermediates that are commonly used in chemical synthesis. Boronic acid are used in Suzuki reactions, Suzuki coupling reactions. So there's a whole series of boronic acid specifications that have been established. And you can go through each of the major types of chemical reactions that medicinal chemists use when they're synthesizing a drug, and the reagents and the intermediates that are part and parcel of those different types of reactions have their own lists of specifications for the chemicals that are associated. But what about mutagens and carcinogens? This is an area where there's a great deal of, I would call it misperception in the public arena around how much of a mutagen or how much of a carcinogen you can safely be exposed to. most people would say little or none, which is accurate. But the none part is really not something which is defensible scientifically. And I would just offer as an example, thinking about mutagens and carcinogens. There was a paper published a number of years ago, which estimated that the number of DNA adducts, or number of DNA damage sites in each cell of our bodies on any given day is about 50,000. So there are about 50,000 DNA damage sites within each cell of our bodies every day, whether we eat healthy foods or unhealthy foods, whether, regardless of how clean our lifestyle is, we live in a world where we're under assault 24 hours a day by chemicals that are damaging our DNA and contributing to the aging process. And by and large, our bodies are evolved to defend ourselves against those chemicals and their insults. Some of them, though, do get through and contribute to long-term adverse health effects. But my point here is we don't live in a perfect, clean world. It's not like we live in the Garden of Eden anymore. So when we think about how much of a mutagen or how much of a carcinogen can be present safely in a drug substance or a drug product, we have to bear in mind that we don't live in a perfect world. And so to help us reach some decisions about what some of these mutagenic and carcinogenic substances, what levels we could be exposed to, the ICHM-7 guideline came up with the TTC, the threshold of toxicological concern, of 1.5 micrograms per day. Now, where does that come from? It corresponds to a risk of less than 1 in 100,000 excess cases of cancer over the course of the lifetime of the individual. Okay, so it's less than 1 in 100,000 that these mutagenic and potentially carcinogenic substances have to be controlled at. So we're controlling these at less than 1 times 10 to the minus fifth, or 1 in 100,000, against a background risk of 1 in 3. As I said, we don't live in the Garden of Eden. We live in a world where our DNA is constantly being attacked, constantly being damaged. And the background cancer risk is 1 in 3. So if you didn't smoke and you didn't, engage in any other risky behaviors, you just took the human population, the background risk is 1 in 3 persons dying from cancer. So the TTC is intended to control these mutagenic impurities and degradants to a level of 1 times 10 to the minus fifth, less than 1 times 10 to the minus fifth. It's based on a linear extrapolation from a dose giving 50% tumor incidence at the most sensitive site in the most sensitive species. So most carcinogenicity studies are done in rats or mice. There can be tumors forming at different sites, liver, sarcomas in the muscle. There can be blood cancers, lymphomas, or leukemias forming in these rats and mice. So whatever is the most sensitive species, the dose which is associated with a 50% tumor incidence is extrapolated down linearly which is a very conservative way to do the estimate, to come up with this 1 times 10 to the minus fifth. So actually, it's much less than that when you think about the extrapolations that are done here. The TTC is based on a lifetime exposure. And we'll see in the next couple of slides that if the exposure is less than lifetime for a given drug, then the specifications for a mutagenic or potentially carcinogenic substance could be higher. There are multiple conservative assumptions that go into the TTC calculation. So as I said, the actual risks are very much likely, in fact, almost certainly, much less than 1 times 10 to the minus fifth, or 1 in 100,000. If you read the M7 guideline and the other guidelines that it refers to, there are multiple nuances in the guidelines for marketed products. So what I'm giving you here is kind of a very high level flyover. But if you got down into the actual weeds and started trying to navigate, through the guidelines for a specific drug, it's actually much more detailed and much more complicated than what I've said. The assessment for mutagenic and potentially carcinogenic substances has to include looking at both the actual impurities, the ones that you know are there, as well as the ones which might be there. And that oftentimes results in conversations between pharmaceutical companies and regulators, because the regulators obviously are going to err on the side of caution. And also they have a broader awareness of, they see far more drugs from different companies. And so they'll know if one company has a problem, if another company is in a similar situation, the regulatory agency will direct them to assess a potential impurity, even if it hasn't actually been detected yet. There are some classes of mutagens which are excluded from the TTC concept. Aflatoxins, the nitroso compounds, and azoxy compounds are excluded. And the reason for that is because they are known or believed to be far more potent for causing human cancers than what could be controlled with a simple TTC approach. So if something is really a bad actor, it would be excluded from this approach, this 1.5 micrograms per day allowance. Before we get any further into looking at how the categorization or the classification of mutagens is done, I just want to take a moment to point out how the mutagenicity is assessed under ICH M7. Part of the assessment that an impurity or a degradant would have to go through to determine whether it's mutagenic could involve this bacterial mutagenicity test, sometimes called an Ames assay. This is by far the most commonly used assay for detecting mutagens in the world today, and it's probably going to stay that way for quite a while. It's a very simple assay. Basically, what's done is to take some bacterial strains, either Salmonella strains or Escherichia coli. These strains contain pre-existing mutations. So these bacteria already have mutations in them, which have prevented them from synthesizing essential amino acids that they need to live. So the Salmonella strains cannot manufacture their own histidine, and the E. coli strains cannot manufacture their own tryptophan. And as a result, if they are plated on agar or growth medium that does not have that particular amino acid included as a supplement, then they will not grow because they can't make their own. They have to have either histidine or tryptophan supplied as part of their culture medium that they're growing on. So the bacteria are treated with the test chemical. They're plated on, as I said, this nutrient deficient agar. And if the chemical caused a mutation that reversed the pre-existing mutation, then those mutants would be able to grow. They would have regained the ability to synthesize their own histidine or tryptophan, and they could grow in a nutrient deficient agar. And what that would look like, if you looked at the Petri plates that these things are grown on, if the test chemical was not mutagenic, it would look like the plate on top, where you would have a few colonies forming because there's always a few background mutations taking place as the cells divide. But if the test chemical was a mutagen and it did reverse these pre-existing mutations, it would allow a lot more colonies to form, and that would be seen as a positive signal. And there's a vast literature that you can go look up. You can Google Ames assay if you want to read more about this, or you can contact me and I can get you some references if this is something you want to follow up and learn more about. So how does that fit into classifying mutagens and carcinogens when they're known or suspected of being present as impurities in a drug product? So the process is sort of outlined here on this slide. The first step, if you have a known bad guy, a known mutagenic carcinogen, it falls into class 1, and you have to control it at a specified acceptable limit, which has already been established. So if there are mutagens which are known to be carcinogens, then there's a fairly large database already available for them, and the chances are that they would already have a spec established or specification level established, and you would have to control its presence in your drug product at that specification level or less. If you had something which was sort of a new chemical and you had very little data, the way those are handled is with computational tox assessments, or what they call QSAR methods, and these are commercially available. And basically what's done is the chemical structure of the molecule is assessed, and if it has certain chemical groups, aromatic amines or aldehydes or other chemical groups that are known to be associated with mutagenic activity, then it would have what's called an alerting structure. And if it's an alerting structure, then that puts you into category 3, where you would have the choice of either controlling at the TTC, or you could do an Ames assay, as I showed on the previous slide, and if it was non-mutagenic, even if it had the alerting structure, if it was non-mutagenic, you could bump it down to class 5, or if it was mutagenic, you'd bump it up to class 2. Okay. And so just with a very minimal amount of information, you look at the chemical structure and you do an Ames assay, and you can, from those 2 pieces of information, you can come up with a decision about how much or how little of that impurity or that degradant can be present in the drug substance or the drug product. Okay. So this is a way to drive decisions really when you have very little information, toxicity information on the chemical itself, but you're relying on background information from many, many, many other chemicals that have been assessed previously. If the drug is indicated for a less than lifetime exposure, so some drugs you take essentially for the rest of your life. And for the rest of your life, if you think about that, it's whoops, I'm going to do that. If you have a 70-year lifetime, that's 225,550 days. If you have 1.5 micrograms per day spread over those days of your life, then you would have a total lifetime dose of 38.3 milligrams. If your drug was less than lifetime, then you could allow a higher limit or a higher specification for that impurity or that degradant, depending on how long the individual would be taking the drug. And you can see there in table 2, if it's less than a month. I mean, some drugs you only take for a very short time. Some you take for an intermediate length of time, and then some you're on essentially for the rest of your life. And there would be different specifications for the mutagens or the carcinogens, depending on the indication and the anticipated duration of therapy. I don't want to dwell on this too much. This is just meant to capture or perhaps illustrate the importance of the TTC analysis. I mean, why do we even bother to do them? What decisions are being driven by these analyses? I will tell you that the approval, the lot release specifications are reviewed, and each lot it has to be approved for release before it can be marketed. So even if a drug has been approved, every time a new lot is manufactured and comes into the marketplace, they have a little number stamped on the bottle, what lot number it is. Each lot has to be reviewed, assessed, analyzed, and approved. And that is a staggeringly expensive process. And so the TTC analyses are a way to decide which of these options for defining the release specifications for a particular drug can be defended. And so it's very, very, very important, not only from a safety perspective, but also from a speed and manufacturing perspective. And if you really want a good example of how things can go wrong, you can go back a few years ago. There was a drug, Viracept. It was an AIDS drug, and it was found to be contaminated with a carcinogen. And this awareness occurred after the drug was on the market. So it was an AIDS drug, which at the time was life-threatening. It had a huge advocacy group in its patient population. It was a serious contaminant that was discovered after the lot had been released, for the public use. And Roche, the chemical company involved, had to do a lot of work to understand how to manage that issue. And if you're interested in learning more about that story, you can contact me and I can give you the references and you can read up on that. But when things don't go right, it's very, very challenging for the patient population as well as for the drug manufacturer. And that's the last thing we want to see. That's the last thing we want to see when we're trying to help people who are already suffering from diseases and other unmet medical needs. So a couple of closing thoughts here. The Kramer's rules, which the TTC was drawn from, was not invented or conjured up within the pharmaceutical industry. It came out of the food industry originally, but it has been adopted by other sectors of industry as a risk management tool. The TTC has global regulatory acceptance. So it's worldwide accepted by regulatory agencies in pharma. It's very effective at supporting decision-making for chemicals that have not yet been studied. So you can have very little data and still come up with very, very sound, defensible decisions. It's enormously important for enhancing our ability to get the drugs out of development and into the patients who need them. It reduces animal testing so that it's very 3Rs friendly. And I just would add this one note here. I put this in so I wouldn't forget to mention it. For those who are not in the pharma world, you have to understand pharmaceutical companies operate under a risk-benefit ratio. So in evaluating risks, you can always look at the benefits. So if a drug has an indication for a life-threatening disease, then risks can be higher, like a cancer drug. For drugs that have a much less dire indication, then the risks are like zero. Like I worked on a lot of diabetes drugs in my career, zero tolerance for risk, because there's so many other alternatives, alternative therapies that are available. Why would anyone want to come up with a drug that has a new risk? So there's the risk-benefit ratio with pharma. In other industries, like the EPA world, there's risk, but there's no benefit. So if there's something that gets into the groundwater or there's something that's in the air that you're breathing, from the EPA perspective, there's no benefit. It's only risk. And so trying to just apply TTCs across different regulatory sectors, you have to understand that the regulatory agencies may have different ways of thinking about risk and different tolerances for risk. So that's just something to keep in mind. There are other proposed applications for TTCs. As I indicated, EPA is thinking about using these. There's a reference there if you're interested. There are also inhalation and dermal TTCs that people are trying to come up with. Everything I talked about in this seminar was for oral route of administration. There's even some ecological TTCs, but I don't know too much about that. But I know that people in the ecology world are looking at that. And then finally, there are a couple classes of chemicals or materials which are excluded from the TTC concept. And those include inorganics and metals, proteins, steroids, nanomaterials, radioactive materials, and compounds that are very, very highly bioaccumulative, or that have a very high propensity to accumulate either in an individual or in the environment. So a good example of that is the dioxins, TCDD, which accumulates in various mammalian species, but to different degrees. And so it's difficult to come up with a TTC based on animal data when you're trying to extrapolate to humans when there are species differences in the degree of accumulation. So that concludes my seminar. we went just a little bit longer than I had hoped, but I'll stay for questions if anyone has any questions.
Thank you so much, Dr. Reynolds. Really great presentation. I know we had a large number of attendees today, so I'm really happy they were here to hear your talk. So I will open it up to the Q&A. So I know people were interested in having the recordings and if it'll be distributed. So yes, we will have the recordings up after this. We put them on our YouTube channel and then they'll be on the website. So that it just includes the slides as part of the video. If you'd like the slides separately, we can get those to you, but we don't post the actual slides themselves. Okay, so we do have a question. Is the inhalable TTC for gases, or does that apply to aerosols as well?
That's a good question. I've not worked with any inhalation products in my career. As a toxicologist, I could only refer you to the literature references that I provided, and when you look at one of these, look at the references in that reference, and you begin to chase things. It's like, chasing things back up the tree, and you may be able to find some information there. That would be my best advice to you. I'm not at all an expert on inhalation or dermal TTCs. And in fact, even for the oral TTCs, I relied very heavily on a group in my company to help me with this. There were people that are extremely adept at this, they've forgotten more than I'll ever learn. And if you're stuck and you have a question, my email is there. Get a hold of me and give me your question, and I'll put you in touch with someone that can help you.
That's great. Perfect. And so that was one of our trainees, Shruti Shahadri. So I know if she has any further questions, she can definitely find you.
Yep.
And just a reminder, if anyone's trying to ask a question, you have to ask the question in the Q&A field and not the chat. The chat doesn't work during webinars, only the Q&A field.
There's something I see on my screen. There's 2 Q&As.
So those are the ones that I already asked you. So
Okay.
I think we're good there.
So the last thing I would say is that, ordinarily in an academic setting, which I think many of you are in, you have the luxury of time to study a problem. And the idea of making decisions or driving decisions in the absence of data would be something that's very uncomfortable. I know when I started in industry, that was something that I felt very queasy about. But if you look at the pressures that we operate under, and the underlying assumptions which err on the side of safety and err on the side of conservative assessments, so that buffers or cushions are built into the decision-making process to protect against overexposing someone, you can actually reach some very defensible decisions with very, very minimal data sets in a very, very short amount of time.
That's great. We have 2 more questions that came in. I know we're at time, so I'm just going to read through them quickly for you. Okay. TCCs are excluded from metals and bioaccumulative substances. I'm assuming PFAS is under that umbrella. Any thoughts on how TCCs could eventually be developed for these excluded compounds?
I have thought about, and I've actually asked a couple people about TTCs for things like PFASs, and I haven't. I don't have an answer. It's obviously something that people are going to be thinking about. One thing I would say about PFAS is the little that I have read on them, they are not genotoxic. They are almost certainly going to be guided by thresholds in their dose response profiles for cancer, or probably for any other toxicity. And the exposures that an individual would have would probably be mostly from drinking water, I would think. And so you'd have to sort of imagine how much greater the occupational exposure would be on top of the background exposure from drinking water, and where that would fit you on the threshold of a dose response curve. There's no doubt in my mind that the PFASs and other non-genotoxic agents operate by threshold mechanisms in their dose response profiles. Not a shadow of doubt about that.
Okay, great. And then the last question, great talk. Are there studies on bacterial assay related to firefighters that you can tell us about?
Absolutely. I would, if you'd have to send me an email, but going in and doing skin wipes, the urine mutagenicity studies that David DeMarini published, the urine mutagenicity studies with the Ottawa Fire Department that Jennifer Keir and Paul White published. There was a study done of people, believe it or not, working in the charcoal industry. I think it was in Brazil, where they were like inside these smokehouses all day long, working on making charcoal. And then their urine was collected and assessed for mutagenic properties in basically Ames assays. If you shot me an email, I could certainly provide you with some references there that would get you started.
Okay, perfect. I will make sure they have your information. Well, this was wonderful, and we're right at time, a little bit over, so I don't want to keep everybody, but thank you all for being here for our March symposium. We are scheduled for next month, same time. Hegla, can you remind me who is presenting next month?
Egla is on mute. Oh, she's looking.
Sorry, I thought I had it in my back pocket. I don't have it. So I think it may be Dr. Malay on the personal exposure reporting app. But either way, everyone will get a reminder of who
I'm so sorry. I didn't realize I was in mute.
will be presenting.
Yeah, it's Barbara Millay.
Okay, so Dr. Barbara Malay will be presenting for our April seminar. So we are super excited to be talking about our personal exposure reporting app. Also want to take a brief moment just to thank you all for attending in person or virtually our 2024 International Firefighter Cancer Symposium in February. It was a great success. For those who have been waiting for the videos and slides, they should be posted sometime early next week of all of the speakers from the symposium. Again, if you have any questions related to anything regarding the studies or any of the events coming up, please always feel free to reach out to any of us at FCI. Have a great rest of your week, and we'll talk to you soon. Take care.